So let's first delve into the core of our topic, the neuroscience of sleep. And this section will explore the intricate neural mechanisms that govern sleep. So I think many of you've probably seen this. This is a hypnogram showing an idealized breakdown of the stages of sleep through the night. And you can see that human cycles through non-REM sleep and REM sleep. So non-REM sleep. And non-REM is divided into stage one, two, and three, and one and two, and then three. And these stages cycle approximately every 90 minutes in an ultradian rhythm, amounting about 4 to 5 non-REM cycles per night. And there's typically more N3 sleep in the early night and more REM sleep later in the latter half of the sleep period. Now, how do we distinguish these sleep stages? And this is primarily done with the EEG or electroencephalogram, and it's part of a broader some or PSD assessment. And this slide shows the characteristic EEG patterns. So we notice the progression of non-REM sleep as we move from wakefulness with its alpha waves to deeper sleep. The EEG becomes increasingly synchronized. And so stage N1 shows lower amplitude, mixed frequencies, mainly theta waves at 4 to 7 hertz. Stage N2 is defined by sleep spindles, those brief bursts of faster activity, and K complexes, the large distinctive waves, which is biphasic in nature. And then stage N3, which is deep sleep, is characterized by high amplitude, slow delta waves. And REM sleep, however, is completely different. The EEG desynchronizes, resembling wakefulness with its higher frequencies and lower amplitude waves. We also see theta waves during the tonic phase and unique sawtooth waves during the phasic phase of REM. And of course, you have rapid eye movement and muscle atonia, basically a paralysis of your muscles, which is measured by electrooculagram and the EMG, respectively. And these are also crucial for identifying REM. So these distinct EEG patterns are the key to objectively defining and analyzing each stage of sleep. So we go from the patterns of wakefulness and sleep to understanding the underlying mechanism. And this slide introduces the reticular activating system, a critical network for wakefulness and arousal. And the foundation of our modern understanding comes from the pioneering work of Maruzzi and Magoon, which was done in 1949 and pictured here. And their key discovery was that the ascending reticular activating system located in the upper pons and midbrain locations were critical for arousal. And basically what they did was they took anesthetized cats, stimulated this region, and that would shift the EEG of the cat from slow synchronized waves of sleep, just like what we saw, to the desynchronized active pattern of wakefulness, even if the direct sensory input was blocked. And so this demonstrated that the ascending reticular activating system has a powerful intrinsic ability to promote wakefulness. And this diagram shows projections from key brainstem nuclei, releasing neurotransmitters such as norepinephrine from the locus coeruleus, serotonin from the raphe nucleus, dopamine from the substantia nigra and the ventral tegmental, and the histamine from the tuberomammillary nucleus, and acetylcholine also from the tegmental areas of the midbrain. So this widespread activation is what underlies the alert and awake state. Now, this slide illustrates a simplified model of the sleep-wake switch, highlighting the key brain regions and pathways involved. And let's first focus on the wake promoting side shown on the left. And we see the familiar players from our previous slide, the brainstem nuclei, including the locus coeruleus, the dorsi raphe, the basal forebrain, all sending projections upwards. And this activates our awareness and arousal. And we also note important players like the orexin neurons. And these areas collectively activate the cortex and the thalamus, maintaining wakefulness. Now, looking at the right, we see the sleep promoting side. And the key player here is the ventrolateral preoptic nucleus, or VLPO, and also the median preoptic nucleus in the hypothalamus. And the VLPO is kind of like a master switch. It's the sleep promoting centers that generally inhibit all the wake promoting centers to promote onset and maintenance of sleep. So this slide illustrates sort of the flip-flop nature of the sleep-wake switch, showing the reciprocal relationship between the wake promoting and sleep promoting neuronal activity. And this graph plots the firing rates of key neuronal populations across transitions from wake to sleep. On the left, we see the transition from wakefulness to the light non-REM sleep. And you notice the firing rates of wake promoting neurons shown in blue for acetylcholine, gray for histamine, green for norepinephrine from the locus coeruleus, sort of decrease as this animal transitions into sleep. Simultaneously, the firing rate of the neurons in the sleep promoting preoptic area, which releases GABA, is shown in red, increases. The right graph shows the reverse, the transition from non-REM sleep back to wakefulness. And as the animal wakes up, the GABAergic sleep promoting neurons decrease their firing, while the wake promoting cholinergic and histaminergic and noradrenergic neurons increase their activity. So this reciprocal inhibition, where wake promoting neurons suppress sleep promoting neurons and vice versa, is the hallmark of a flip-flop switch. And this is shown here, basically in a much more easy to visualize diagram. And again, you can see that the monoamines and acetylcholine are on the wake side, GABA in from the VLPO, and MNPO is on the right, associated with sleep. Now, this flip-flop model, while useful conceptually, maybe is an oversimplification in some ways. In reality, the dynamics of this neurotransmitters are much more nuanced. And this slide shows actual measurements of the monoamine levels, not just firing rates across wakefulness, REM, and non-REM. And you could look at serotonin, histamine, and norepinephrine. And let's just take a look at the serotonin, for instance. And blue is here marked by wake. The white areas is non-REM, and red is REM. And you could see that the levels of the monoamines are highest during wakefulness and generally lower during non-REM sleep, but they don't completely disappear. Instead, we see a gradual decrease in serotonin and histamine as the animal progresses through non-REM sleep. And this is important to demonstrate that the sleep-wake transition isn't a binary on-off switch for these monoamines. It's more of a dimmer switch, a gradual modulation of their levels. And so far, we presented the sleep-wake flip-flop switch as a relatively isolated system, but in reality, it's beautifully integrated with other crucial regulatory processes. And we still have a basic balance between these two sides. However, now we add orexin, a neuropeptide produced in the lateral hypothalamus. And orexin acts to stabilize the wake state, promoting wakefulness and preventing inappropriate transitions to sleep. And we know that individuals who lack the ability to secrete orexin has narcolepsy, because then they, have this unstable switching back and forth between the wake and sleep. In addition, the entire system is modulated by two major forces. First, the circadian rhythm, our internal biological clock, exerts a time of day dependent influence, promoting wakefulness during the day and sleep at night. And second, the homeostatic static sleep drive, which builds up with prolonged wakefulness, primarily through the accumulation of substances like adenosine. And adenosine acts on A1 receptors to increase the sleep pressure. So these modulators act on both the wake and sleep centers, and it's a dynamic balance, constantly adjusted by internal factors like orexin and external cues conveyed by the circadian and homeostatic drives. And so this is just a quick summary of the representations here. During wakefulness, the monoamines, acetylcholine, and orexin is elevated. During non-REM, GABA is elevated. All right. So now we've established the basic flip-flop switch between wakefulness and sleep, which is primarily driven by the balance between the monoamines and acetylcholine and the GABA neurons. Now let's add another level, the switch within sleep between non-REM and REM sleep. And notice the lower portion of the diagram, we have a second seesaw representing this non-REM-REM balance. During non-REM sleep, as we have seen, the VLPO and NPO are active, inhibiting the wake-promoting centers. But within this state, there's also reduced activity from the REM-promoting cholinergic neurons of the PPT and LDT in the brainstem. Norepinephrine and serotonin contribute to sort of go against this REM sleep. And so the transition to REM sleep often involves a shift in this balance. Activity decreases in the noradrenergic and serotonergic neurons further, and there's a surge in activity in these cholinergic PPT-LDT neurons. And this cholinergic drive, combined with the continued inhibition of the wake-promoting centers by the GABA, results in the unique state of REM sleep, which is, represented by desynchronized EEG, muscle atonia, and dreaming. And so we have a nested system, a primary flip-flop switch between wake and sleep, and a secondary flip-flop switch within sleep, governing the alternation between non-REM and REM. And so this slide sort of details the brain circuitry behind the non-REM sleep switch. We have REM on regions like the cholinergic LDT and PPT, and REM off areas like the ventrolateral paraqueductal gray and LPT, which are GABAergic. Orexin from the lateral hypothalamus, I don't see that here, it suppresses REM, while MCH or melatonin concentrating hormone may promote it. So crucially, the ascending projections from the LDT PPT are responsible for the key REM EEG features, such as theta waves, via connections to the septohippocampal region. And the descending portion from the SLD, another part of the REM on areas, cause muscle atonia of the REM sleep by inhibiting motor neurons, primarily using GABA and glycine. And so this sort of provides a summary of that. You have the monoamines and acetylcholine elevated during wakefulness. You have GABA elevated in both stages of sleep, but then you have acetylcholine and MCH elevated during REM sleep, with some minor activation of monoamines during the non-REM sleep. And so this slide builds on our previous look at monoamine levels, now adding acetylcholine dynamics across the sleep stages. And we've already seen that serotonin and histamine are highest during wake, decrease in non-REM, but have levels in REM. And now if you look at acetylcholine, this shows a biphasic pattern. It's high during wakefulness, as expected, but it decreases significantly during REM, non-REM, but crucially, it increases again during REM sleep, reaching levels compared to or even exceeding wakefulness. So you may be wondering, like, why am I spending so much time on these neurotransmitter systems and the flip-flops switches? It's because I think it's relevant to how many of the common medications affect sleep and wakefulness. And let me give you some examples of this. On the wake side, we see that amino inhibitors increase monoamine levels, which can enhance alertness, but worsen insomnia. Antihistamines like mirtazapine or doxepine block histamine, a key wake-promoting monoamine, and this is why they're used often as sleep aids. On the sleep side, we have benzodiazepines and alcohol, with both enhance the effects of GABA, and with thus promoting and strengthening the sleep side of the switch. And if you look at the non-REM balance, like SSRIs and many antipsychotics, this increases the norepinephrine and serotonin that favors non-REM and then also suppresses REM. And finally, Alzheimer's disease, characterized by a loss of cholinergic neurons, often leads to significant deficits in REM sleep as well. And so this slide illustrates the connection between our sleep centers and a broader central autonomic network, as explained by the neurovisceral integration theory. As you can see, sleep centers are primarily located in the hypothalamus, midbrain, and pons region that are also linked to the stress, motor, control, and behavior and the endocrine system. So this anatomical overlap underscores that sleep is not just the process of shutting down. Instead, it's deeply integrated with the autonomic nervous system, which is the system responsible for regulating heart rate, digestion, and stress response. And so the neurovisceral integration theory posits that these brain regions collaborate to coordinate our internal body state with our behaviors and environmental responses. And this integration is bidirectional. We have bottom up influences where the signals from the body, such as stress hormones and pain, can affect sleep. But there's also top down influences where practice like meditations, which can impact the prefrontal and cingulate cortex, can modulate the activity of these lower brain regions and in turn influence sleep. So in essence, sleep is a dynamic pattern process influenced by both influences on both bottom and top down factors.